Good morning, everyone. Can you hear me? Yeah. Welcome to the first plenary session today. My name is Audrey Duncanson. I'm from the Wellcome Trust in London. And similar to Cindy's comments yesterday, we've been involved in the Global Alliance from the very beginning, and we're very proud to be involved in such an important global initiative. It's my pleasure and privilege to introduce Heidi Rehm as a plenary speaker this morning. Heidi is well known to many of you in the audience from GA4GH, but for those of you who don't know her as well, Heidi is from the Broad Institute, where she is the medical director of the Institute's clinical research sequencing platform. She is also the chief genomics officer at the Centre for Genomic Medicine at Mass General Hospital and a professor of pathology at Mass General, Brigham and Women's Hospital and the Harvard Medical School. Heidi is a world leader in driving the standards and best practice for the interpretation of sequence variants, and as the principal investigator of ClinGen, making sure that this information is shared widely and openly for the benefit of patients around the world. Heidi also co-leads the Matchmaker Exchange, which we all know is a driver project of GA4GH that allows the sharing of genetic data for patients with rare disease. As we've just seen in the video, the sharing of such data across multiple platforms really maximizes the chances of finding the candidate gene, which we saw is hugely important to the patients and their families and can actually lead to clinical intervention. Heidi is a pioneer and a dynamic force for open science and data sharing, and we look forward to her talk today, which will focus on advancing medicine through data sharing and collaboration on a global scale. Please join me in welcoming Heidi to the stage.
Thank you. It's been a great meeting, and thank you to everyone for coming for the last few days. So I'm going to talk about some of the projects that we've been involved in, really trying to advance medicine through data sharing and collaboration really around the world. I'd like to open my talks with real personal stories. I shared, I think, one about a pregnancy termination that could have been prevented with data sharing in one of the last Global Alliance meetings. Today I'll mention a paper that we were involved in, really highlighting patients who have been misdiagnosed because of lack of access to global population data, and this one related to patients who were misdiagnosed with hypertrophic cardiomyopathy due to lack of non-white population data that could have been used to rule out variants that were assumed to be pathogenic. And I remember one story of a freshman basketball player who had a full scholarship, first person in his family to go to college, and he got a full basketball scholarship, but he passed out on the basketball floor, led to a cardiac workup, had a slight thickened heart, which could either be physiological hypertrophy due to his athleticism or the early stages of hypertrophic cardiomyopathy, and we did testing on him, and we found a variant of uncertain significance and had to report that to that basketball player. He was taken off the team, lost his scholarship, and his, probably only shot at college. So that was, a traumatic story. I think now we know that variant is benign because of population data. And actually most of the variants that we see when we do genetic testing are in fact benign, but we don't always have the ability to say that. And this map from this one particular paper, you can see here that a variant, let's see if I can show that doesn't work. All right, well, anyway, if you look at the US, you'll see a particular blue circle that has no color in it because it's a variant found in that population that's incredibly rare. But if you look at the tip of Africa, you can see that variant is actually present in nearly a quarter of those individuals. We can easily rule it out as pathogenic, and that is because of global data sharing. So, again, I've made this argument many times in my life, but for the best outcomes for patients, we really need to enable data sharing on a global scale and critically evaluate evidence as a collective community. I'm now, thanks to my longtime colleague Daniel MacArthur, moving to Australia. Mark Daly and I will be taking over the Nomad project, and I'm happy to announce that we released 71,000 genomes aligned on Build 38 just last week. 700 million variants now released. This has been a tour de force of a lot of people involved, both 140 PIs that have agreed to share their data from 63 major projects, and 80 people that are involved in the Nomad team getting this data up onto a server for access to the community. So a really great resource, but we still have challenges. The computational infrastructure for large-scale variant calling QC is still incredibly challenging, having watched the evolution of the last call set over the last few months. And surprising to me, even though Nomad data sharing is incredibly easy for the PIs, we basically email them and say, hey, your data was already generated at the Broad. Would you mind if we included it in the aggregate allele frequencies on the Nomad website? No one will have access to your individual level data, et cetera. And there are still people who say, I don't really want to share it. And there's nothing we can do. So as much as I love coming to this audience where everyone is very pro data sharing, we still have work to do in the community to convince others that just including your data in an aggregate allele frequency is not that risky. We also are challenged because there's no phenotypes. When I go into Nomad and I find that variant in one person, I just want to know what's their phenotype. And we aren't able to share that. Some of that is willingness, but actually a lot of it is regulatory barriers and consent, and that is work that we as Global Alliance need to help overcome. I will say in a future release of Nomad, we're planning to put links to those samples in dbGaP and later Anvil, so that if there is access to that phenotype, you will be able to find that sample. So that'll be a great addition. We are still, as much as that incredibly colorful figure there, UMAP plot showing all of the diversity in Nomad, we still are missing a lot of genetic diversity from Africa, Asia, Middle East, Oceania. So there's still a lot of data to be shared. And all of the data in Nomad was generated at the Broad. It is very difficult for us to take outside data in and enable it to be combined with the data at the Broad. This is where the Global Alliance comes in really critically to make equivalency in our pipelines and allow data from all around the world to be pulled together in resources like Nomad. So that's really generating population data. As was mentioned, I'm one of 9 PIs involved in the clinical genome resource, where we are really trying to engage the global community in curation of variants and interpretation of those variants and creating public resources, which we host both on clinicalgenome.org as well as working with the ClinVar team at NCBI. So there is a huge number of people leading this project. These are just a slide with our work group, our PIs, steering committee, work group co-chairs, but there are over 1,125 people around the world involved in this project, as shown here, from 30 countries. So really, we've really been pulling people from all around the world to get involved. We have time zone friendly calls, although you may be at various hours of the night. It's very similar in Global Alliance, I'm sure you're all familiar with. But it is a venue to also train many new curators. And ClinGen is really a consortium of people developing standards and curating knowledge. ClinVar is where we deposit the variants that we curate. And you can go to the ClinGen website and see all of the different expert panels that are either formed if the green bar is full or in the process of forming if it's still partly. If you want to volunteer, and this is actually a great way to get individuals trained on variant and gene curation, is to have them join an expert panel. They interact. We both train them through a lot of webinars and online tutorials, and we have YouTube videos, but then they're interacting with the experts around the world as they work on curation and really learn it well. So if you're interested in volunteering, you can go to the ClinGen website, fill out a survey, and tell us what disease area you're interested in. So one of the areas we curate in is gene disease validity, and this helps inform genetic tests and which genes are appropriate to include them. You can see on the figure on the right that there are lots of tests out there that have entirely different sets of genes in them because there hasn't been a standardized approach to looking at the evidence to date. And so we developed a semi-quantitative approach to evaluate evidence that would inform then the inclusion of these different genes and different types of tests depending on the level of evidence. And these are just some of the 814 genes that have been curated to date. Shown in red are those genes that have been reported as disease-associated but really have insufficient evidence. And it's about a third of genes that we evaluate based on their reports for disease association that have insufficient evidence to really be able to use them clinically. So this is an important area of curation for our field to really critically evaluate that evidence. And that, we have different sort of results across different disease areas. I think the most profound one was Brugada syndrome, where 20 out of 21 genes were disputed as involved in disease despite being reported as such. Now, how are clinical labs actually implementing these standards? So this is an example of Invitae's website where they changed their panel. It now has the one gene that we showed had sufficient evidence, and the rest you have to manually add on as preliminary evidence genes if you want those tested. So this is just an example of really trying, as a global community, to improve the quality of clinical test offerings. And you can go to our website and download all of the curated genes if you wish to use them in your own pipelines. For those of you in the National Initiative session yesterday, you heard from Ellie McDonough, Zornitsa Stark, and myself around a wider collaboration beyond ClinGen that we are working on through the Gene Curation Coalition, which started with all of the logos on the left of those groups that had public online resources for gene relationships. We've now, on the right, started to include a lot of other labs that have large curated data sets they've been generating internally but haven't ever shared them. You can see some of the numbers of genes curated in Invitae, 14,000 genes. They're willing to actually share them with us and with the global community. So we're now working to harmonize, and we did a Delphi survey to harmonize the gene disease validity terms across all the different resources, ending up with the final set on the right for those terms. And then the GEL team with their panel app has agreed to develop an instance of panel app where we can bring all these different curations together. And this is just an example of one gene that has been has 10 reviews. This is a demo version that's actually available on the link. I'm happy to share these slides as well. And so that, we'll bring together both the panel app that Australia just launched this week, I think, the Genomics England panel app, and then this shared instance that we're working on with GenCC, all accessible and shareable with data across them. And then you could open up that and see all the different curations in the system. Right now it's color-coded, but we'll be moving to the standardized terms that were agreed upon through the GenCC Delphi survey process, and be able to reconcile differences in interpreting gene-disease relationships. We've also been generating variant resources, both through common standards in terms of terminology, and we have a work group really building off of the original ACMG guidelines to develop those standards that have now been widely adopted. But on the ClinGen website, we have many different additional documents for more granularity around those standards, as well as gene and disease-specific standards coming from these expert panels. They're all publicly accessible on the ClinGen website to help with really finessing the variant interpretation process. But even when we create standards, we still sometimes do things differently, interpret those standards differently. And so it's really critical for accurate variant interpretation to share your interpretations. And so we've been working with NCBI for the last, it's been probably 6 years now, to have everyone be able to share variant interpretations through ClinVar. There's over 888,000 submissions now and over 500,000 variants coming from 72 countries as of the stats last night. So an enormous resource, and what we can now do, because everybody's sharing their interpretations, is we can look at where we differ. And if you go into ClinVar, you'll see that there are 6,312 variants that have medically significant differences, so that patients would be treated differently. That's a high priority area of focus for our work groups. And we actually have a team that's been working between the labs to resolve these differences in interpretation. In the first phase, we had 87% resolution by the labs all sharing their data and reevaluating all those variants. We've tried to make that more efficient through an outlier approach, asking just the lab that differs from all the others to first evaluate. That resolves less, but it's much less time consuming. So, and even if we don't even facilitate this, just simply by sharing data in ClinVar and seeing what others are interpreting and seeing their evidence, it resolves about 24% of differences over time. So really a great way that this sort of global data sharing is improving the accuracy of variant interpretation. Now, we don't expect 100% concordance. There are areas of variant interpretation that are subject to professional opinion. The most common area we differ is evaluating functional data and whether you trust that assay, whether you think it was validated well enough. So that is where we bring in experts who are more familiar with these assays and how they work and whether they're appropriately validated. And that allows us to bring up the variance from individual labs up to this 3-star status that overrides the others through our expert panel process. And we now have a number of expert panels that are approved through ClinGen. We also obtained status recognition as a FDA-recognized database from the FDA. That was a year-long process going through that with them. But that enables companies now to use our interpretations directly in their IVD device and other approvals for implementing test platforms. This is just an example of 10,000 expert classified variants in ClinVar that resolved 758 conflicts through that expert interpretation process. So really helping get these over the finish line. Now, we don't just resolve conflicts. In fact, we target variants where multiple labs have called them uncertain significance, because it turns out if you get all those labs together and you share all that data, you can often move those variants either up to pathogenic or down to benign. And this is just an example of a variant where that happened, where the expert panel worked with the clinical labs, got the evidence. And in fact, up in the right corner were another 7 variants that went through this. So a lot of these variants are actually through sharing the underlying evidence, being able to be classified more usefully than that basketball player that got the VUS back. So we also are challenged with the genes. As I mentioned earlier, about a third of these genes have insufficient evidence. Some of these may just have a little bit of evidence and need more. So how can we take some of these genes that have a little bit of interesting evidence, but maybe move them up the ladder? And that's where the Matchmaker Exchange, a demonstration project, early demonstration project, the Global Alliance, comes in this concept where we can computationally bring data sets together. Here, we're trying to share both candidate genes that have been flagged, the same gene in 2 different cases, and bring their phenotypic data to score the degree of match based on phenotype. When we do this, there is dual notification of the submitters, both the person whose data was sitting there for a while and then the new person who came in and matched with it. That allows equity and both groups being able to be notified when these matches occur and then collaborate to publish those findings. So this platform was launched now several years ago and has become the Matchmaker Exchange, which I think is a great example of a successful federated platform, taking data sets and databases that were really structured very differently in different countries, but by working with the Global Alliance to define a common API and a common set of even just a small number of fields that we use in this matchmaking, that was implemented across all of these nodes so that we can exchange those candidate genes, bring matches together, and build evidence for gene causality. And we worked with both the regulatory and ethics work group around the consent requirements, what was prior to the actual discovery work stream, but the equivalent of it around the API, and the ClinPhen group back then around the sort of disease terminologies and ontologies that are being used. So to date, there's data from over 100,000 patients in the Matchmaker Exchange across all of the different nodes. Shown below is data from GeneMatcher at the Baylor Hopkins group, where the number of genes has just been steadily increasing and the number of matches to those genes also increasing. And data is coming now from over 84 countries around the world. There's really, at this point, no group that works in rare disease gene discovery that is not using the Matchmaker Exchange. So I think it's a really great example of just an incredibly useful platform for this particular case of trying to build evidence for gene causality. We've been using it at our Broad Center for Mendelian Genomics, where we've sequenced now over 10,000 samples. We continue to discover novel genes through this process, largely because of the Matchmaker Exchange. Just give you one case here of a recent case that's still actually just recently been discovered. This was a patient with a variety of clinical features shown there, and we found a canonical splice site variant in a gene that had never had a disease association, GA4GH. It seemed to destroy a splice acceptor site. The variant was absent from NOMAD. The gene was intolerant, but we didn't actually have both parents to show that it was de novo. This is certainly not enough to implicate this gene in disease. And so we would be stuck at this point. But we put it into Matchmaker Exchange, and we matched with a group that actually had 20 patients with CNVs and putative loss of function variants in this gene. There was reasonably good phenotype overlap across with our patient and the others that enabled us to secure the diagnosis. This publication is in process, and we have returned these results to the family. So, yet there's nothing out there on this gene right now in the public domain. So this is just a great example of bringing this evidence together to move these genes to give useful results back to families that are desperate for a diagnosis. Now, this is an example of what we call two-sided matching. So both groups have to have identified the gene as a candidate based on that rare mutation that we saw, for example. But we'd like to move this on to a world where we can do one-sided matching. Maybe I have a candidate, but I think there's somebody's case out there that is not looked at it carefully enough to find a match. But if I query all your raw data, maybe I can make that match. So we are starting to work, and this is work done both within the Matchmaker Exchange as well as with the discovery work stream, and Ben Hutton has been leading work on the case discovery API to really launch a platform. There's the first node of what we hope to be a future federated platform was launched by Ada Hamash's group, the Variant Matcher. And here I can just put a variant in, which I did in this case. On the right are the results that came back from Variant Matcher from the case I entered. In this case, my patient had the features at the top: hypotonia, microcephaly, global developmental delay, and esotropia. I got back a list of cases, half of which were unaffected. The others had phenotypes that did not match. I was able to quickly rule this variant out that I thought was a candidate in my case and be like, nope, not a candidate, move on. So, as I mentioned earlier, most of the variation we see is actually not disease causing, but trying to rule it out is really difficult sometimes, and this is an example where I needed that phenotype to really rule this out. So we're really working, as I mentioned, Ben has been leading this on development of this case discovery API to access raw data for matching and ruling things in or out, and some of the groups that have been involved in this shown here. We have been just in the last few days really discussing how this does or doesn't overlap with the Beacon platform and trying to combine these in useful ways, and that's an example of really this community getting together and sharing use cases and trying to figure out how to most effectively and efficiently solve these challenges. So we'll continue to work on this and getting platforms launched. But then sometimes we don't have any candidate in our cases. In fact, a lot of the time we look at cases and we don't find anything in there that we can interpret. So how can we start to bring our data sets together and share them in ways that ideas can surface? And so then I'll mention a couple of platforms. So the Anvil Platform that was recently funded that really brings together a large number of groups that are contributing to this really space for both data sharing and analysis. And this is, no surprise to this group, but really moving from this traditional approach of having to download data into your own environment to look at it, to really bringing researchers to the data in a cloud-based approach. This is built on the Terra platform that the Broad Institute has launched recently in beta version in April. And it really enables a platform for both data sharing, implemented on the Google Cloud platform. It is FSMA compliant and allows us to put patient data up there on the platform and basically bring large data sets that have been generated across many projects together with analysis tools that are all up on the platform to facilitate community access to the data as well as in situ analysis. And so the types of data that are being shared on Anvil include sequencing data, raw files, metadata, VCF files, as well as a lot of work on getting richer phenotype that has compared to what has traditionally been shared in other spaces. And so this is a work in progress across each consortium to define all of the rich phenotypic data that the groups want to share. There's a phenotype work group for Anvil, but it's also collaborating with Global Alliance, Clin Phenotype, particularly Robert Carroll, who's been leading the Anvil phenotype work group, but also working with many others here through that. We've actually put all of our broad rare disease data up there. You can see a screenshot of that data, which has detailed phenotype, affected status, gender, even variants that we have identified as causative or suspicious, HPO terms, all of that there, really enabling rich access and analysis to the community. I will give one case that's already happened because we stuck our data up there, and a colleague in Utah went and used the data and accessed it, and in fact applied some new structural variant calling algorithms and found a structural variant that we now feel is causative for this patient, diagnosed with GA4GH syndrome, and that result is going back to the family. And that is not because we actually discovered it, but because we shared our data and let anyone else look at it. And we've now validated that structural variant with other structural variant calling algorithms. It is indeed real and impacting the gene. So a great example of enabling diagnoses for patients by just making your unsolved cases shared with the community. So another major program where many of us are working on is the All of Us research program, trying to gather data from over 1 million people. And to date, there's over a quarter of those anticipated participants already enrolled from 399 sites in the US. Huge emphasis on underrepresented in biomedical research individuals, 81% of that status, 52% racial and ethnic diversity, really trying to get a very diverse cohort to enable globally relevant research. On all of those participants, data is being collected in terms of participant-provided info, electronic health records, physical exams, and biospecimens, and that data is being brought into a data and research core that is a collaboration between Vanderbilt, Verily, and the Broad Institute to really get that data, organize and clean it up, and then make it accessible to the community. Working with the Global Alliance Dury work stream in developing the data access approaches, many of which you're hearing about from the work groups yesterday, to enable both public, registered, and controlled access depending on the sensitivity of that data, and allowing very broad access to the data through this more passport approach. Now, there's lots of data types coming in, EHR data, surveys filled out by the participants, these PPI, participant-provided information, physical measurements, Fitbit, and other electronic data, and there'll be new data types added over time. There's a huge quality cleanup process that the Vanderbilt and Verily teams are doing to make this high-quality data that can be usefully used by the community. And I got these slides from Robert Carroll late last night, so thank you to him. This is just stats that he sent me last night on what's been collected. So you can see over 200,000 participants have filled out one or more of these PPI modules, participant-provided information. In terms of the EHR data, we now have over 110,000 records coming in, as well as 190,000 physical measurements. So all of this data being aggregated, which will be incredibly useful. And this platform that I mentioned, built on Terra, allows even someone who's not technically savvy like myself to go in and create a cohort, identify different attributes, and do analyses and statistics right in situ. So really enabling a much broader use of this kind of data to a wide variety of individuals. Also in the All of Us program, one of the key areas that I've been involved in is generating this sequencing data, which we're still working on due to holdups of the FDA. but we're getting there. And we will also return medically relevant results to the participants. There'll be both an arm for ancestry and traits that'll come off the arrays. We've been able to do that now outside the FDA because it won't be health related. And then we'll focus the whole genome sequencing on the return of medically actionable results once we get FDA IDE approval. But to do that, we have 4 different clinical labs involved in the interpretation of variants and returning results. The 4 you see here, Color, Baylor, UW, and LMM. And so we have to harmonize and make sure every variant is interpreted the same across all of the 1 million participants. So we have been working on a project to harmonize. We ingested almost 50,000 variants in just these 59 genes across the 4 clinical labs. Actually, we were able to try to harmonize them. We needed to use some standards. So we used the ClinGen allele registry that uses the newly approved Global Alliance VR standard. So delighted with that success story. And then from those 50,000 variants, 23% were interpreted by more than one lab so that we could compare the interpretations. We were 98.5% concordant across these 4 labs at the reporting level. For the 1.5% that were discordant, 0.86% were medically significant differences that we are working on resolving. And so now we have a discrepancy resolution work group working in concert with ClinGen to resolve those variants so that before we launch this platform, we will be harmonized for all previously reported variants, and then we'll have an interface to then, in real time, ensure there's no discrepancy between the 4 labs as we individually report out these results. So a real effort in harmonizing our processes and sharing data. And this data is also then shared in ClinVar. So I think that's my last slide. I just want to acknowledge many of the programs that are part of the work I presented, as well as including the Global Alliance and funding resources at NIH that have helped support these projects, as well as the Broad Institute. And I'm happy to take any questions. Thank you.
Thank you very much, Heidi, for that amazing talk. Just a quick reminder that you can submit some questions through the Slido interface. But I thought we'd probably start with some questions from the floor while people are thinking about what to ask. Any questions?
Nephi Walton with Geisinger. Just one question on ClinVar. I feel like one of the best resources for information about phenotype and variants comes from clinical geneticists, and there's not really an incentive there for them to contribute in any way. A lot of the work that goes into providing phenotype information with variants goes into publications, which don't always make it, and you'll get things much farther down the road than if they just contributed, while they were seeing the patient and making an interpretation. Has anybody discussed or thought about any kind of incentive or making that part of the academic evaluation process to get providers more involved?
Yeah, it's a great question. So we have been working with both the American Board of Medical Genetics and the NSGC to create maintenance of certification modules for them to actually get involved in evaluating variants of uncertain significance and contributing to that and submitting those to ClinVar. So providers can, and of course Geisinger is a great example of a group that has been having clinician submission to ClinVar to inform variants that labs have prior reported but can be more richly interpreted with the phenotype data. And Stanford has also done that. So you are correct, there's still very few participant-provided interpretations in ClinVar, but we are trying to incentivize that in some way. Now, that said, I do think one of the best ways is to communicate back to the clinical laboratory and ensure that data gets back to the lab so that they can update their variant that's been submitted to ClinVar, as opposed to having a conflicting one come in from the clinical provider. So, I think both things are approaches that are there, but my ideal approach is sort of that teamwork with the clinical lab. And unfortunately, the clinical labs do have slightly more robust processes for submitting to ClinVar, which allows the physician to not have to worry about that process. But I do agree with you, the more that we can incentivize either sharing back to the lab or direct submission to ClinVar is important. And we will continue to focus on that. So it's a great question.
Great. Well, there's quite a few questions there. I don't know if you can see them, Heidi, on Slido.
What global alliance standard is currently most critically missing for these data sharing projects? Oh, there's so many. So it's a great question. I think just because my mind is on the ClinVar topic, we are thinking about, is there a way, because right now you can't submit to ClinVar by API, and I know a lot of groups would like to do that. And so thinking about a standardized API for knowledge sharing around variants, I think would be useful, but then we actually want the platform to do that along with it, so that's on the implementation side. I do think gene annotation, so the Genomic Knowledge Standards Work Group has just launched VR, working on variant annotation as the next standard, and then I'd like the gene annotation to be the next one after that. So I think that's one. I could probably come up with many, many more. Certainly standards around harmonizing variant calling pipelines so that we can bring more data than just Broad's data into resources like Nomad would be important areas to standardize. So I'll just stop with those few, but are there people working on implementing Terra on AWS and other clouds? I might have to defer that question to someone in the audience who is working on Terra. Is anyone out there jump in on that? Or maybe we can wait to the break. Let me move on to something I could answer. What is ClinVar's plan to reevaluate pathogenicity decisions now that we know Nomad and GAT call sets undercall some homozygotes? So just one critical thing, ClinVar does not evaluate pathogenicity. They are a database that enables external submitters to share their interpretation. So it's actually getting the submitters to reevaluate. So we are doing a lot of that work through ClinGen expert panels, through the variant discrepancy resolution, and going back to some of these submitters to say, gee, here's a list of variants that are clearly not pathogenic because they're at high allele frequency. So ClinGen has been managing that, and all GAT calls it's undercall some homozygotes. Actually, this is a thing that's been recently discovered in Nomad around these homozygote calling, and so the team is actually working on flags in Nomad for some of this calling issue, and that will be fixed in another iteration, so that there's two different things going on there, is the accuracy of zygosity calls in the Nomad dataset release, as well as variant interpretation. When ClinVar has conflicting interpretations, does it report both? Yes, so ClinVar reports everything any submitter says, and if they're different, the overall interpretation is conflicting. Now, when expert panels come in and evaluate that, because they're 3-star, they actually override, so the overall interpretation is going to be a single thing from the expert panel. However, it's important to continue down because all of the submitters' interpretations are in there still. And the ClinGen expert panels try to keep things up to date, but new data comes around all the time. And if a new clinical lab reports something differently more recently, you might want to pay attention to that. So they're all there. You can look through them. And if you find a variant in a patient, look at everything. If they're all old, they're probably out of date. But it's important to do that. And we have reports that we send all the expert panels for any new clinical lab interpretation that differs from the expert panel so they can rapidly evaluate and contact that lab for that data. So that's how we're trying to deal with that, but it's an ongoing work effort. What things should Global Alliance put on our roadmap to better address some of the challenges? Okay, I'm just gonna, in the interest of time, I'll make a list. But there's, I named some of them when we were talking about standards. But I do think, if the community could come together around an API-based exchange of variant knowledge from some of the databases being developed, like the Sharient in Australia and GEL's database, we've been having some of these conversations, but we just haven't had the resources to bring together an environment for API-based sharing that we could then perhaps export and also put in ClinVar, but have that real-time access. I think that would be a project that Global Alliance might be able to facilitate in terms of standards. Can we assume a variant with high minor allele frequency in one ethnic group is benign in another ethnic group? So largely when you're talking about rare disease, we usually can make that assumption. Common disease, it's totally different, particularly when you've got a tagged variant that is not actually the causal variant all bets are off. They are very ethnically relevant. That said, as we think about the variation in penetrance, incomplete penetrance is undoubtedly due to modifiers, and they may be racially specific modifiers. So I think over time, it will be important to think about that and what other variants might impact the phenotype. But I will say, if you find a very high allele frequency in a population that's large enough and does not have an astounding high prevalence of the disease, for rare variant interpretation, you can largely rule that out. Although, very subtle minor effects probably can't be fully ruled out. When discussing Nomad, you said that some contributors are reluctant to contribute data for aggregation. What reasons are given? Well, one group, I have been trying to get a reason and haven't yet, just the committee voted. That was the only reason given. For another group, the answer was, I really want my postdocs to have first access to those variants. Okay, no comment there. So I would say not good answers. This is where the funders in the room can have a little more say in this data sharing approach. What Matchmaker results will affect ACMG classification? Matchmaker right now, let me think about this. So for the variant matching that I gave an example of, that case from Variant Matcher where we got back 8 cases of well-phenotyped individuals because they were studied as part of rare disease diagnosis, and all of those individuals did not have the phenotype I was looking at, you can use that as observations in unaffected individuals, which are part of the ACMG guidelines for classification. That said, it's a little tricky because those rules are based on genes that have been already implicated in disease. It's a little trickier to think about these rules when there is not yet a disease implication. So you really think about damaging and other sort of attributes. So those things do go hand in hand, but there's some trickiness to it. Will ClinVar or ClinGen use phenopackets? So that right now, there's very little case-level data being submitted to ClinVar, but I do hope that that more groups will submit case observations, which you can. It's on a separate tab in the Excel spreadsheet. But what I brought to the speed dating session on Monday with the ClinPhen group was we need a tabular implementation of phenopackets, and that will make it easier because right now people submit to ClinVar with a tabular spreadsheet. And so if the fields for phenopackets were written out in a tabular form, I think it's more likely that submitters would actually adhere to this standard. And so we did talk with Melissa and David and the team about a tabular implementation of phenopackets, and I think that would make it easier. What are the data protection techniques that are used and envisioned in Matchmaker? This is actually an interesting question. We've been having this discussion because when we launched Matchmaker, the data security standards were really not developed. This was one of the first demonstration projects. So now, one of our requirements for participation was adhere to the Global Alliance data security standards. And so new nodes are now coming in. We're like, well, you have to do this. And they're like, whoa, there's a lot there. And so we have been having discussion. We actually, in the speed dating with the security group, had some discussion around the type of data sharing we're doing, which is a gene and HPO terms, and the security needed for that type of sharing versus individual level data. And maybe we need a slightly different version that is specific to our project. So I think thinking about this and in terms of the risk and benefit and environment and what you're sharing, I think is a nuance that we're going to need to work on so that we have appropriate data sharing for the level of risk and the data type. So more to come on that topic. Does your system have clinical accreditation? So there is no clinical accreditation in the way we clinically accredit clinical labs for systems like Nomad, Matchmaker, et cetera. I don't know which system they're talking about. But the ClinVar, the variants classified by ClinGen expert panels have, as I mentioned, this FDA-recognized database status. So that could be used directly in clinical care and IVD devices to the FDA and things like that. The Anvil Platform, I don't know which system is being asked here, that is FSMA compliant, and they are working on really having it compliant according to CLIA CAP standards as well, which are in some ways similar there. So many of these systems and the All of Us research program, that is all CAP accredited CLIA certified, with one exception: that samples are not fully CAP and CLIA compliant at the draw, but all the other processes are. So that answers a few of them. You guys can stop me at any point when you need me to get off the stage. These go on.
Yeah, we've actually run out of time.
What? Oh, no. Oh, you can put them back up. If there's more time, I'll keep answering. At some point, my leg will collapse. But, okay, we'll do 2 more. How can we encourage database to which hold this data to prioritize and fund the development of... Encouraged. Trying to read that sentence. I'm going to say fund the development of sharing resources. So I'm just going to say that we can encourage by funding. Grant mechanisms that really help build platforms. Because one of the challenges with Matchmaker Exchange is that all of these groups, there was no common source of MME funding, and so everybody sort of volunteered their time. And updating the system and adding new features has been a challenge for us in that area. So, a dedicated grant for that particular focus, I think these are things that will help. Has there been any progress with GEL sharing their data for Nomad? This part goes to a question that I mentioned, or an issue, which is right now Nomad really takes only data from the Broad, and that's around this equivalency issue. It is very cost and labor intensive and difficult to bring outside data in, and it comes back to standards and equivalency of pipelines. So this has not been tackled, not because of GEL being unwilling to share data in this particular question about Nomad. It is because we need a better mechanism to be able to harmonize the data so that they can be brought into common resources. So there are other issues there, not just willingness.
In the interest of having tea, I think probably you should stop and give you a break.
Ewan was encouraging me to go on.
I know. Please join me in thanking Heidi for her amazing talk.